morning my dear student. Welcome in the SWAYAM course Genetic Engineering and its recent advancement. Myself Dr. Tanushree Chatterjee from Raipur Institute of Technology welcome you all. Today I will be discussing about transcriptional regulation in prokaryotes. Why gene regulation? Why we regulate gene? For developmental changes, cell specialization, adaptation to the environment and prevent creation of unwanted product. So, what is the purpose of the gene regulation? To respond to their environment, it is quick that saves lot of energy. The basic concept of the gene regulation at the transcription level in prokaryotes can be explained with the help of operon model that was proposed by Francois Jacob and Jacques Monod in 1961. He received Nobel Prize in 1965 and he worked on genetic control of enzyme and virus synthesis. So, these are our Nobel laureate. They received Nobel Prize for their work in physiology and medicine in 1965. Jacob and Monod distinguished between following two types of sequences in DNA. First is sequences that encode for transacting products, usually protein, and cis-acting DNA sequences. Gene activity or expression is regulated by the specific interactions of the diffusible transacting protein products with the cis-acting DNA sequences. In prokaryotic genes and operon, genes involved in related functions often clustered together and expressed as a unit on a single mRNA, that is operon or polycistronic mRNA. Gene expression must respond to environmental conditions. Some regulatory proteins present in 5 to 10 copies per cell, some enzymes for glycolysis present at 1 lakh copies per cell. Genes whose products are presently unneeded or acceptable levels are turned off. Mechanism to achieve proper mix of gene expression are varied, whatever works best strategy. Now, genes and regulatory elements, structural genes that encode protein, regulatory genes encode products that interact with other sequences and affect the transcription and translation of these sequences. Regulatory elements, DNA sequences that are not transcript, but play a role in regulating other nucleotide sequences. In constitutive expression, continuously expressed under normal cellular conditions. Positive control that stimulate gene expression, in negative control inhibit gene expression. So, operon is a complete and coordinated unit of prokaryotic gene expression and regulation. So, what is operon? Operon is a complete and coordinated unit of prokaryotic gene expression and regulation. Now, what is lactose hydrolysis? Enzyme encoded by LAC-Z gene, glucose and galactose products, enzyme cleavage broad range of beta galactosides including synthetic analogues such as ONPG and X-gal. Operon include three things: a set of structural gene whose products are required by the prokaryotes to complete a metabolic, that means catabolic or anabolic pathways. Second one is a regular gene whose product is required to regulate the expression of structural gene. Third one is control element, promoter and operator. So, the operon structure is promoter additional sequences that control transcription plus structure gene. In regulator gene, DNA sequence encoding products that affect the operon function, but are not part of the operon. In this pictorial representation, you can see the process. Lack operon expression. In absence of inducer, repressor tetramer binds to operator and blocks RNAP from binding to promoter. Now, negative and positive control, that is inducible and repressible operons. So, in inducible operons, transcription is usually off and needs to be turned on. In repressible operon, transcription is normally on and needs to be turned off. Negative and positive control. Negative inducible operon, the control at the operator site is negative, molecule binding is to be operator, inhibiting transcription. Such operons are usually off and need to be turned on. So, the transcription is inducible. Inducer, a small molecule that turns on the transcription. Now, negative and positive control, that is repressible operon. An additional control system is superimposed on the repressor operator system. This system exists because cells have specific enzymes that favor glucose uptake and metabolism. If both lactose and glucose are present, synthesis of beta galactosidase is not induced until all the glucose has been utilized. Thus, the cell conserves its metabolic machinery that, for example, induces the lac enzymes by utilizing any existing glucose before going through the step of creating new machinery to metabolize the lactose. Studies indicate that in fact some catabolic breakdown product of glucose, no exact identity is yet known, prevents activation of the lac operon by lactose. So, this effect was originally called catabolite repression. This is the process you can see in pictorial representation. Here also the negative and positive both. Now, E. coli lac operon. LAC1 has a constitutive promoter and is expressed separately from the lac operon that encodes the lac repressor and low level of expression. Lac operon expression. Lac operon encodes polycistronic mRNA giving rise to 3 different enzymes. Gratuitous inducers. Lactose is normal inducer, actually allolactose for the pure at heart, but other B galactosides also work. Isopropyl thiogalactoside IPTG also act as an inducer, but is not metabolized. Shows induction does not involve interaction with the actual enzyme being synthesized. Isopropyl thiogalactoside IPTG gratuitous inducer not metabolized, at level remains constant once added. The lac regulatory mutations. In constitutive mutations, lactose repressor gene LAC1 mutants, an operator region mutant, that is OC. Various combination of these and other mutations studying using F plasmids encoding all or portion of lac operon, that is merozygotes. IS mutants. Mutation eliminates induced binding site. In IQ mutants, high level quantity expression. Here also you can see very clearly. In IS dominant mutations, this is the summary. If we see the observation, adding glucose to lactose medium reduces lac operon expression by cells dramatically. Another level of expression, cells must use most efficient sources of carbon or energy. Glucose is most efficient because all necessary enzyme expressed constitutively. CAMP synthesis, CAMP means something to virtually all cells. For E. coli, it means I am hungry. Synthesized from ATP by adenylate cyclase. Now, catabolic repression, catabolic activator protein CAP, also called catabolic regulatory protein or CRP. Positive control of catabolic operation, allosteric, that is regulated by CAMP, ATP up, CAMP down, then ATP down and CAMP up. CAMP, CAP binds to promoter and it converts relatively weak promoter into one of the strongest in the E. coli system. Here you can see the CAP in incorrect conformation to bind promoter when not bound with CAMP. CAP in correct conformation to bind promoter when bound with CAMP. Auxiliary operator region, 2 additional repressor binding sequences located, maximum repression when all bound, binding O1 and O3 by a repressor tetramer, 2 DNA binding site causes looping of DNA similar to that found in eukaryotes. Lac repressor and operator, you can see the crystal structure here. TRP operon encodes 5 structural genes whose activity is required for tryptophan biosynthesis. The operon encodes a single transcription unit which produces a 7 kb transcript. The TRP repressor, gene product of TRP-R operon, specifically interacts with the operator site of the TRP operon. TRP repressor is a dimer of 2 subunit. Only when the tryptophan binds to the repressor, it will bind to the operator of TRP operon. Tryptophan, the end product of the enzymes encoded by the TRP operon, therefore, act as a co-repressor and inhibits its own synthesis through end product inhibition. The repressor reduces transcription initiation by around 70-fold. In E. coli TRP operon, it encodes 5 polypeptides required for tryptophan biosynthesis. Anabolic operon TRP EDCBA has repressor like lac operon, but called aporepressor, that is inactive by itself. Tryptophan is the co-repressor, repressor active only when bound with tryptophan. Knockout TRPR genes and operon is still repressed in the presence of tryptophan. E. coli TRP operon, no TRP present here, you can see here very clearly. Transcriptional regulation through attenuator. Initially it was thought that the repressor was responsible for all the transcriptional regulation of TRP operon. It was later observed that the deletion of a sequence between the operator and TRPE gene coding region results in increased level of the both basal and activated, that is de-repressed transcription. This site is termed as attenuator. The attenuator, this site lies end of the transcript leader sequence. The attenuator is a RHO independent terminator site which has a short GC rich palindrome followed by 8 successive U residues. If this sequence is able to form a hairpin structure, it acts as a highly efficient transcription terminator. Then only a 140 BP transcript is synthesized. The leader sequence contains for 4 regions of complementary sequence which can be formed different base paired RNA structures. The attenuator is the product of the base pairing of sequence 3 and 4. Sequence 1 and 2 also form a second hairpin. However, sequence 2 is also complementary to the sequence 3. If 2, 3 form a hairpin structure, then 3, 4 attenuator hairpin will not form. Leader peptide encode 14 amino acid, 11th and 12th codon of this leader RNA encode successive tryptophan residues. The function of leader peptides is to determine tryptophan ability and to regulate transcription termination. Tryptophan is a rare amino acid. Therefore, under the condition of low tryptophan availability, ribosome would be expected to pause at the site. Transcription and translation are highly coupled in E. coli. So, translation can occur while mRNA being synthesized. The ability of the tryptophan, the ultimate product of TRP operon, is sensed through its requirement in translation of leader peptide. In the presence of high level of tryptophan, the ribosomes proceed along the passage slightly behind the site of transcription. Under these conditions, the mRNA regions designated 3 and 4 hybridized to form a stem loop structure that signals the termination of the transcription in the presence of low level of tryptophan. However, the ribosome stall at region 1 of the mRNA, which consists 2 adjacent codons for tryptophan. In this case, since region 2 is not bound to a ribosome, it is free to form an alternative stem loop structure by hybridizing to region 3. This hybridization prevents formation of 3 to 4 stem loop and transcription is able to continue past the attenuator sequence. Transcription factors are protein that regulate the transcription of gene by binding to corresponding regulatory DNA regions, usually localized in the proximity to the transcriptional start site of a gene or operon, often designated as promoter, that is 1 and 2. Depending on the position of this DNA binding site, the TF can act as an activator or repressor for the target gene. There are many databases covering transcription factors, TCS or signal transduction pathways in prokaryotes. P2CS11 contain TCS of all available bacterial and archaeal genomes and their classification. The Swiss Institute of Bioinformatics TCS databases contains a large data set of predicted TCS based on a Bayesian model. The MECIATS database contained a large collection of signal transduction pathways for a large amount of organisms. KEGG14 also provide a TCS pathway map covering TCS regulated genes, while these databases contain a comprehensive overview of available TCS and TFS various organisms. None of them provides the binding site of the corresponding response regulations. BioPsych aggregates biological data for thousands of prokaryotic genomes from various databases as well as to form manual annotation. This includes a large amount of regulatory data including TF binding site, position on the promoter region. To integrate as many as bacterial transcription factor binding site as possible, Prodoric was established in 2003 containing transcription factor binding sites from 5 organisms: E. coli, B. subtilis, Pseudomonas aeruginosa, Listeria monocytogenesis and Helicobacter pylori. With the 2005 update virtual footprinting, a tool for in silico regulation prediction was integrated into ProdoNet, a tool for identification and visualization of gene regulatory networks based on data stored in Prodoric or generated by virtual footprint, was released in 2018. The latest release of Prodoric in 2007 started the overhaul and modernization of Prodoric to bring the database to a state of art visual and technological standard for future development. Here we finally conclude this transition process of the almost 2 decades old database to present a modern, easy to use and highly effective bioinformatics tools. Prodoric now distinguishes between the 3 types of experimental evidences for a DNA binding site of a transcriptional regulator, which are in vitro expression evidence, physical protein DNA binding evidence and binding site variation evidence. Correspondingly, different methods for every evidence group was defined. Those evidence groups are based on evidence and conclusion ontology. In vivo expression evidence describe the ability to change the expression of a gene when the transcription factor of interest is bound to the corresponding promoter region. In vivo methods sustaining this experimental evidence group include any kind of reporter, gene based expression, assay like glucuronidase activity assays, beta galactosidase activity assay or fluorescence based reporter assay. Here the promoter region containing the transcription factor binding size is cloned to the reporter gene and the expression is either measured with and without transcription factor. Importantly, result from solely high throughput methods. Regulation phenomena to the database. Nevertheless, the automated solid interpretation of such data with respect to the corresponding underlying gene regulatory networks will be subject to future prodoric development. Physical protein is a DNA binding evidence. Describe the actual physical binding of transcription factor to specific segment of DNA. In vitro methods including footprint assays or gel retardation, electrophoretic mobility shift assays are widely used to detect the binding of regulator to a specific segment of DNA. Common methods are nasal footprinting assay, methylation protection interference assay, 10-phenolphthalein copper footprint and hydroxyl radical protection footprint. The binding region is either defined by the protected area of the footprint assay or by the DNA fragment successfully used to gel retardation assay. Binding site variation evidence describe the verification of a actual DNA binding motif of a transcriptional regulator of interest by site directed mutagenesis or successive deletion of the binding region in order to define an exact location and DNA sequence composition of the actual DNA binding site. Both techniques are usually combined with in vitro expression assay, footprint analysis of EM assays. For every binding site, the experimental method was extracted from the literature and is displayed on the matrix. Additionally, a circular evidence indicator with 3 segments representing the 3 evidence group. Then sigma factor. Sigma factor are the environmental signal specific DNA binding subunits of RNA polymerase and essential for transcriptional initiation. They mediate environmental signals including nitrogen status, heat and extreme heat shock, various environmental stresses, the need for flagella formation or iron transport. Sigma factors bind around the 35 to 10 regions of a classical bacterial promoter near the transcriptional start site, while some sigma factors need a specific spacer region length between conserved 35 to 10 motifs, or other sigma factor tolerate different spacer length. Prodoric sigma factor are stored as one of the database entry containing the aligned conserved 35 to 10 region separately by a variable spacer region. Most sigma factor binding sites are defined by experimentally determined transcriptional site using the primer extension technology and therefore may not have experimental evidence as I described. Thank you all.